Hello, hello. Okay. It's great to see everyone. We're at 01 now, and I'm sure more people will join us. We just wanted to say thank you for coming. Welcome again. We'll let people continue joining as we get started today, but we are finally at our second edition of the semester's Black and Count Bio seminar series. We're really excited to have Dr. Derrick Morton join us today from USC. And so basically, Dr. Morton's research interests are basically studying the fundamental mechanisms of post-transcriptional regulation or gene expression with an emphasis on RNA processing factors mutated in human disease. Specifically, they're interested in the post-translational transcriptional activities of the RNA regulatory Rna Exosome complex in human neurological disease. They've taken the strategy of coupling in vitro molecular with biochemical studies with in vivo Drosophila genetics and multi-omic approaches to understanding these different aspects of post-transcriptional regulation of RNA. So the focus ranges from defining tissue-specific roles of RNA processing, surveillance, and decay machinery to how defects in essential and ubiquitous RNA processing factors cause neurological disease. Dr. Morton received his BS in biology from Eastern Kentucky University and his PhD in biology from Clark Atlanta University. So we're really excited to have him here today to share a little bit of his work as well as his journey. So Dr. Morton, welcome and take it away.
Yes, well, and thank you for inviting me to come talk with you all, speak to you all today. And so as the host mentioned, my name is Derrick Morton. I'm an assistant professor at USC in molecular and computational biology. And today I'm going to talk to you a bit about my journey in academia, but also the research interest of my group. And that is really focused on this molecular machine that's very, very important for fundamental RNA processing and degradation events in the cell, and it's linked to a very severe neurological disease. And so first, I'd like to start out with my journey in academia. I'm originally from Lexington, Kentucky, and I did my bachelor's degree at Eastern Kentucky University in biology. And then shortly thereafter, I went to Clark Atlanta University and did my PhD in biology there in Atlanta, Georgia. And then after finishing my PhD, I did my postdoctoral training at Emory University in the labs of doctors Anita Corbett and Ken Moberg. And so many years later, I am now a tenure track assistant professor at the University of Southern California in molecular and computational biology. And I also have a visiting associate appointment at Caltech in the Department of Biology and Bioengineering. And so a bit about my seminar before I move forward. I'll start with a bit of an introduction, going over post-transcriptional regulation and the importance of ribonucleases in this process. And then I'll get into my current research. And I like to box my research into the fundamental biology aspects of the RNA exosome and its tissue-specific requirements. And then also thinking about the etiology of disease. And so the RNA exosome linked neurological disease, Ponto cerebellar hypoplasia. And I should have mentioned this, I actually opened my lab in August. And so I like to talk to you all a bit about what are some of the future experiments that I'll be doing in my lab following my postdoc. And so I'd like to start out with a key question that's really fundamental to biology. So how does one set of genes give rise to such a diverse array of cellular functions, right? And so every cell in our body has the exact same DNA blueprint, but how is it that we end up with such a diversity of cell types with unique function that are working in concert with each other? And so to begin to answer that question or to understand how that's possible, we can start with the central dogma of molecular biology that really explains the genetic flow of information. And so most of us on this call are very well aware of the central dogma, DNA to RNA flowing to RNA via transcription, and then ultimately getting proteins via translation in the cytoplasm. But still, how does this one set of genes give rise to a diverse array of cells with unique function. And that really is the differential expression of these genes that confers distinct functional properties to each cell type. And so the differential expression of genes allows for us to have the diversity of cells that work together to maintain homeostasis and ultimately support life. And so I forgot to mention, I know there are members of the public here, so I'm going to try my best to make sure everyone's engaged, and we'll walk through some fundamental processes along the way. And so while much of this differential gene expression occurs at the gene-specific transcriptional level, there are numerous post-transcriptional regulatory events that also contribute to cell specific expression patterns, which also dictate function. And that is really where the crux of my research lies, within understanding a co- and post-transcriptional regulation of RNA. And so before an RNA molecule is deemed competent to go out into the cytoplasm, where it is eventually translated and made into protein, there are a series of steps that have to occur co- and post-transcriptionally to ensure that this mRNA molecule is stable and protected from ribonucleases and can be translated properly. And so mRNAs undergo capping, splicing, and continued processing and packaging and export before it is actually translated. And these steps are very, very critical to the overall regulation of gene expression. And we really think about these co- and post-transcriptional regulatory events when thinking about how we regulate gene expression. However, often we don't think about, well, what happens to an RNA once it's lived a very productive life, it's been translated, but is no longer needed? Or what about a defective RNA? And so decay mechanisms are also very, very important to the regulation of gene expression, and often left out of the conversation when thinking about the regulation of gene expression. And so I want to spend just a little bit of time talking about mRNA decay. And in this slide, though it's very busy, I like to show the slide because it really underscores the complexity of mRNA decay. The cell has evolved several mechanisms to turn over RNAs that are no longer needed or defective. And so here I showcase 3 of these pathways that are highly utilized to turn over RNAs in a very regulated, quality control regulated driven manner. And so there are several enzymes, ribonucleases, that are important for these processes and actually turning over and degrading these transcripts that are no longer needed or perhaps were misprocessed or defective. And so today I'm going to focus on a key player in these processes, these processes of turning over RNA in the cell, which is the RNA exosome. It is a key player in also RNA processing, but plays a huge role in the turnover of RNAs as well. And so for the crux of my talk, I'll talk about how the RNA exosome plays a key role in the overall regulation of gene expression. And so before I get into the structure of the RNA exosome, its function, where it's located, I want to pose a larger question of why this is important or why we care about this. And so the complexity of the transcriptome poses a great challenge to cells, right? And so cells must manage RNA processing events, but also getting rid of surplus or defective RNAs, all without destroying functional RNA molecules. And so how does the cell actually manage these events? And so remarkably, the RNA exosome performs these 2 divergent RNA processing roles in the cell in processing of RNA, but also functional maturation of RNAs and processing, but also quality control driven destruction of RNAs as well. And how the RNA exosome actually facilitates or mediates these 2 divergent roles is still very much a mystery. And so in my group, in part, we will be examining the fundamental biology of the RNA exosome and other ribonucleases as well. well. But a bit more about the RNA exosome specifically, it is a multi-subunit ribonuclease with 3' to 5' activity. It is ubiquitously expressed, which hold on to that because that will become important. So that means it's in every cell of your body and is essential. And so likely why this complex is essential is probably because we speculate that it's critically important for the production of mature, properly processed ribosomal RNAs for the ribosome. And so it does have a processing role in other small non-coding RNAs and some mRNAs as well. But as I mentioned, the RNA exosome plays a critical role and is a key player in turning over RNA. So aberrant or misprocessed RNA, and also normal turnover of RNA. So I mentioned once an RNA molecule has lived a productive life and is no longer needed, there has to be some regulated turnover of these transcripts, and the RNA exosome does participate in normal turnover as well. So as you see here, the RNA exosome partitions between the cytoplasm and the nucleus and has functions in each compartment of the cell. And importantly, was first identified in a screen for ribosomal RNA processing mutants in budding yeast. And it's termed RRP actually in lower eukaryotes such as yeast and flies. And that will become important as the talk continues. And so a bit more about the structure. The structure is conserved, and so it is 10 essential and conserved core subunits. The RNA exosome is made up of a catalytically inert core, which is made up of a 3 subunit cap ring, and also a hexameric ring, which is made up of a 6-member ring that makes up this catalytically inert core. And it is thought that RNAs are actually threaded through this core to the one ribonuclease that associates with the bottom of this complex, which is DIS3. And so as I mentioned, this is a ribonuclease, and the catalytic component of this complex is DIS3, and it is a processive 3' to 5' ribonuclease with exo and endo ribonucleolytic function. And so, as I mentioned, it is thought that RNA substrates are threaded through this catalytically inert core down to the catalytic DIS3 subunit. And so I mentioned that the complex was conserved. So what do I mean by that? And so what I mean by that is that the complex is conserved from lower eukaryotes all the way to humans. And so from yeast to flies to humans, the RNA exosome is conserved in structure and function. So it's important for processing and degradation of RNA molecules. And so we do have the current structures of the RNA exosome molecules in yeast and in humans. So here we have the current structure of the budding yeast exosome complex. It's an 11 subunit structure. And as I mentioned, it's termed RRP for the ribosomal RNA processing mutant screen that was done in yeast, such as RRP, and that's in lower eukaryotes, such as yeast and flies. But also we do have the current structure of the human RNA exosome plus 2 cofactors. So here you see the human RNA exosome structure, but you also have different cofactors that are in this structure as well. And it is thought that cofactors actually are delivering substrates to the RNA exosome for processing and degradation of RNA. And so importantly, it's also thought that these cofactors also aid in restraining the RNA exosome function. So you don't want the RNA exosome, a very powerful ribonuclease, actually running rampant through the transcriptome, chewing up all your RNA. So it's thought that these cofactors play a critical role in delivering substrates to the exosome, but also restraining exosome function as well. And here, the structural biologists that solved this structure of the human RNA exosome used a very elegant study to demonstrate the exosome actually interacting with substrates. So they made a DNA RNA hybrid that actually stalled the RNA exosome so they could capture an RNA substrate actually in the RNA exosome being processed. And so here you can actually see an RNA substrate being threaded through this core down to the catalytic subunit DIS3, where it will then act to process and or degrade that substrate as well. And so importantly, in humans, the components of the RNA exosome are termed exoSCs for exosome components. And so lower eukaryotes, just a reminder, is RRPs, and in humans, it is termed exoSCs. Okay, so now we went over the fundamental biology of the RNA exosome, and it's an important post-transcriptional regulator of gene expression. It has critical function in the cell and is very, very important and essential for viability of our cells. But why do we care, right? And so as recently as 2012, for the first time, this ubiquitous complex that, as I mentioned, is a fundamental and essential for RNA metabolism was linked to a very tissue-specific disease, a very severe neurological disease called pontocerebellar hypoplasia. And when this was found, it was quite surprising, given this fundamental role that the RNA exosome plays in RNA metabolism. Why would this ubiquitous complex that's essential for these fundamental roles in RNA processing and decay give rise to this very severe neurological disease? And so here highlighted is pontocerebellar hypoplasia, which is what I'm going to focus on today in my talk. But I also wanted to kind of give some a more kind of insight into the RNA exosome and how fascinating it is in thinking about biomedical science and health in general. And so now other subunits of this RNA exosome have been linked to other diseases, most of them neurological in basis, but also some of these other subunits have been linked to very distinct disease, which is now interesting that why is a different subunits in one structure, one complex giving rise to very distinct disease. And so towards the end of my talk, I'll talk a bit more about that and some of my interests going forward in my lab. But first, what is pontocerebellar hypoplasia? And so pontocerebellar hypoplasia is an inherited genetic disease of autosomal recessive inheritance. So that means there are mutations on both copies. And individuals with PCH1B often have an early mortality as early as 8 weeks. And so here in this cartoon, we have the cerebellum depicted, as well as the pons. And also we have radiographical images of individuals with PCH and normal. And we can see a normal cerebellum and pons, but in the individual age matched here with the cerebellum and pons, we see the wasting of the cerebellum and also wasting of the pons. And so the pons is involved in your autonomic control, which is important for control of breathing. And also your cerebellum is important for control of muscle movement, balance, and coordination. And also in these individuals with PCH1B, they were also reported to have developmental delays and intellectual disability. And so again, in setting this up, why is a ubiquitous complex that's in all of your cells in your bodies, fundamental for RNA metabolism when mutated, giving rise to a very tissue-specific disease that's quite severe? And so now I'm going to move into my current research, and I'll talk about the fundamental biology of the RNA exosome first and some of the experiments we did to examine this fundamental biology. And then I'll get into the etiology of disease and using the Drosophila model to actually model some of these patient mutations. But before I move forward, I wanted to put this slide up to underscore the importance of mentoring and training. And so in my postdoc at Emory University in Anita Corbett and Ken Moberg's lab, I was able to train several students at different stages in their career, from high school to undergrad to graduate students. And I really wanted to impress upon the crowd today that at any level, you can contribute to science and make meaningful discoveries. And nice for for me and these students that they actually got to co-author a paper with me, my most recent article, examining the Rna exosome in Ponto cerebellar hypoplasia in flies. And so I wanted to shout out a person I worked really closely with, Binta Jallo, or let me excuse myself, Dr. Binta Jallo. She is now a postdoc at the National Cancer Institute and worked very closely with me while I was doing this project at Emory. And so getting to studying the Rna exosome and first thinking about the fundamental biology of the Rna exosome, we wanted to understand why is it that the Rna exosome is giving rise to this very tissue-specific disease. It's affecting neurons. It's in all your cells of your bodies, but why is it affecting neurons specifically? And so the first thing we did, we wanted to utilize a system that would allow us to examine tissue-specific function of genes. And so in thinking about the human body, we want to think about the fundamental biology and how can we understand the basic biology or gene function. And so we use the Drosophila model system, which is a very, very nice model system, excellent model system to understand tissue-specific gene function. And so we are actually using the Drosophila model to get at these tissue-specific questions that we have, but also I want to highlight that there are very productive mechanistic studies being carried out by several labs and budding yeast that are asking questions about the mechanism of action of the Rna exosome. What are the fundamental mechanisms of the Rna exosome function that are very productively being carried out in yeast? And so before I jump into some of the data, I wanted to set this up that first, we wanted to understand, well, what were the requirements for this subunit? So we understand that this subunit is the culprit, right, for this very severe pontocerebellar hypoplasia, but it had not been, at the time when we started this study, been determined, well, what were the requirements for this subunit? And so what we did by using a binary expression system in the Drosophila model that allows us to overexpress things and then also knock things down, we can actually take away or knock down gene expression of this Rna exosome subunit that is termed RP40 in flies. And so what we first found is that in knockdown of the Rna exosome subunit gene RP40 that encodes this subunit of the Rna exosome, that when we deplete that subunit, we do not get viability of the flies. So this means that the Rna exosome subunit RP40 is required for viability of the flies, okay? And so we're doing this to really understand what are the requirements in specific tissues and cell types. And here we did a few different tissues where we were able to knock out expression of this RP40 subunit to understand what were the tissue-specific requirements from neurons to glia to muscle and global depletion. We were able to knock down this subunit and there were no viable flies, underscoring that the Rna exosome subunit RP40 is required for viability. And so, well, that's not fun, right? So if you have dead flies, you have nothing to experiment with. And so a cool technique or utility of the fly is that we can time when we knock down certain genes. And so we exploited the Drosophila life cycle to knock down the expression of the RP40 gene 24 hours after egg laying. And so the flies were able to mate, lay eggs normally. And then what we did is we used the temperature shift allele in the background of these flies to modulate when we turned on this knockdown or RNAi to knock down RP40 in these flies. And so at the restrictive temperature, knockdown of RP40 occurs. And so at 24 hours, we shifted these flies to this restrictive temperature, which allowed for us to turn on the RNAi and deplete the RP40 subunit. All right. And so after doing those experiments, we had some surprising data. So in the global depletion, even 24 hours after egg laying, we still had no flies. And so, and to back up from the last set of experiments, that really established that there is an embryonic developmental requirement for the RNA exosome RP40 subunit that is required for viability. And so now we're moving post-embryonic development and trying to test to see if we can get viability. And we did just that. So in moving past that post-embryonic phase, we were able to get viability of neuronal depletion, glial depletion, and muscle. So we were able to deplete in the same manner in which we did in the initial experiments and get viable flies after we waited 24 hours after egg laying. And so our next question after getting viability of these flies, well, how healthy were these flies? Did these flies have any issues? Were there problems with these flies? And so one thing that we did is that we did survival lifespan assays where we actually just looked for whether the flies are, how long they live. And so one thing that we immediately noticed that after eclosing and developing into an adult fly at day 1 and day 2, the one flies that were actually depleted for the RP40 subunit in neurons actually had very, very short lifespan. They only lived for about a day or two compared to all the other controls and depletions in muscle and in glia, which suggested for the first time that there may be a higher threshold requirement for the RP40 subunit and exosome in neurons. And so we followed this up. And so we wanted to then understand, well, we've made it past this post-embryonic developmental requirement for the RNA exosome subunit RP40. Now, are there any neuronal morphology or functional issues? Is the RNA exosome required for normal neuronal morphology and function? And so we wanted to look at the fly brain. And so here I have an image of a fly brain compared to a human. And obviously the fly brain is not structurally analogous to the human brain, right? However, it is functionally analogous. And what do I mean by functionally analogous? So over 80% of known disease-causing genes in humans that we know of are actually conserved in flies. And so what we can do is we can use these lower eukaryote models, such as the fly, to understand the gene-specific function of the RNA exosome. And that's exactly what we did. And so we utilized the fly brain to understand whether or not there was any neuronal morphology or functional issue when we deplete the RP40 subunit. And so we utilized a structure in the fly that is not required for fly viability to test our hypothesis that the RNA exosome is indeed required for normal neuronal function. And so when we use the mushroom body, which is the seat of learning and memory of the fly, and it's characterized by these alpha and beta lobes that are bifurcated, as well as gamma lobes, we were able to understand initially whether or not the RNA exosome played a role in neuronal function. And so this actually gave us a biological venue to test and to do these experiments. And so what happened once we actually depleted the RNA exosome in these mushroom bodies? And so what we found as compared to control and looking at this cartoon, that there were defects when we actually waited 24 hours post-embryonic development and looked at the brains of these flies. And so in looking at the brains of these flies, we actually saw neuronal morphology defects in these flies. And that is defined by the crossing over of these beta lobes and thinning of these alpha lobes. And so in these neuronal populations, neurons have signals to tell them exactly where to stop and how to form. And so here was our first evidence that the RNA exosome is required for proper development of an essential structure in the fly brain when seeing that the beta lobes and alpha lobes had defects. And so kind of to wrap up the fundamental biology section and some of these foundational experiments really testing for the fundamental requirements of the RP40 subunit linked to disease, we were able to find that it was essential for development, which actually was not surprising. Other studies in mice have shown, and in budding yeast have shown, that this component of the RNA exosome is indeed required for viability. However, what was much more interesting is that we found a requirement for RP40 in neurons when we timed this post-embryonic developmental delay of depleting the RNA exosome, so in this age-dependent like manner. And so also we found that the RP40 was indeed required for development of an essential structure for learning and memory in the fly, showcasing for the first time that there was indeed a role, an essential role in neurons for the RNA exosome. And so now I'm going to kind of shift gears a bit, still sticking with the RP40 subunit, but now I'm going to talk about disease. And so in disease, what's happening is that there are missense mutations that are encoding these amino acid substitutions in conserved regions of the RNA exosome. So all of the studies that we've done thus far are depleting the subunit of the exosome and in a post-embryonic timed way, but that is not modeling disease. We are not losing the actual subunit. These are missense mutations in genes that are encoding this subunit that are giving rise to disease. And so again, we're using the Drosophila model to really examine the tissue-specific function of the RNA exosome by modeling these amino acid substitutions that are found in human patients. So here is the domain structure of the XOSC3 gene, a protein in humans as well as in flies. So it is well conserved with identity and similarity. And as you can see, there are a number of missense mutations that are encoding these single amino acid substitutions in highly conserved regions of the XOSC3 gene that have been linked to pontocerebellar hypoplasia. And so how did we go about actually modeling these genotypes in flies? And so we used CRISPR-Cas gene editing technology where we actually created the mutations and knocked them into these flies. And so what we did is we essentially took the missense mutations that we know encode these single amino acid substitutions and incorporated those into the fly genome at the locus via CRISPR-Cas9 gene editing. So now we have flies that are actually modeling genotypes that are found in patients. And so, as I mentioned early on, that this disease is inherited in an autosomal recessive pattern. So both parents are carriers, and then you can end up with an affected offspring or child, in this case, an affected daughter that has hits on both copies of the chromosomes. And what we are doing is essentially modeling this in patient flies. And so to kind of layer on this or to complicate things a bit more, what clinical journals and reports are now reporting about some of these genotypes from patients that they are sequencing, some of these genotypes are correlated to more severe disease or more moderate disease. So there is a disease severity linked to these amino acid substitutions. And so what we did in flies, we took a severe genotype and a moderate genotype and modeled this in flies. And so you'll hear me talk about a G11A amino acid substitution being severe and a D87A amino acid substitution being more moderate. And then also we can put that over a null or a deletion and get a severe phenotype as well. And very importantly, when we design these experiments, we actually CRISPR'd in a wild type RP40 locus. And so all of our flies that have undergone this CRISPR mediated process are being compared to wild type controls that have also been CRISPR'd in a normal or wild type copy of RP40 into the locus as well. And so our first question after we got our flies, and now we're trying to see, well, are there any phenotypes? Are we able to actually make the homozygous and heterozygous genotypes of these flies? And so what's really nice about the Drosophila model is that you can make these flies to create the true heterozygous and homozygous genotypes found in patients. And so for members of the audience that may not be in science, we often are using these model systems to do these things to model disease. So we can't mate humans, right? We can't investigate and examine humans. So we utilize these lower eukaryotes to understand fundamental genetics and how when something goes awry causes disease. So we're interested in the basic molecular and cellular processes that are gone awry, and we're using these lower eukaryotes to do just that. And so in flies, we have this ability to create these heterozygous and homozygous genotypes. And what we found immediately was that we could actually get flies that are modeling the patient genotypes. And what was surprising is that in our viability studies and just understanding the viability, are they alive? Are these flies, are we able to create these models? We were actually seeing that in the actual genotype-phenotype correlation that I talked about in the prior slide, that the correlation of viability with our flies was actually correlating with what the clinical reports are suggesting about these mutations. And so in our very severe mutant, it was very sickly and only had about a 14% viability, whereas our more moderate genotype had a higher rate of viability at about 38%, which is aligning with what is being reported in clinical journals. And so after we were able to determine that indeed these flies are alive, we wanted to then test, well, what is the life expectancy of these flies? And in the same manner, we were able to find that that the severe mutants were actually living shorter lifespans as compared to the more moderate genotypes. And so here you see the more severe D87 over this deletion had the shortest lifespan as compared to G11A, which is also severe, which lived for about 12 to 14 days, as compared to the more moderate genotype, which actually lived longer for about 25 days or so. And so we were able to actually demonstrate that when we engineered these patient mutations and cross the flies to model these genotypes, that we did have impaired viability and also shortened lifespans that actually was on par with disease severity that is being reported in clinical reports. And so next we wanted to see, well, are there any kind of gross morphological phenotypes with these flies? And indeed, we did find that the flies did have wing phenotypes. And in the fly world, if you will, we often look at the Drosophila wings to understand, are these flies able to behave normally? Are they able to fly? Are they able to walk normally? Because the fly wings are important for that, and also often model neuromuscular injury to the fly. And so here we find that the wings in an age-dependent manner had wing phenotypes. And so this is very important that we actually are seeing in an age-dependent manner that we are finding phenotypes in these flies. So at day 0, the flies are perfectly normal, but as the flies age, we see these phenotypes, and that is quantitated here. And so I wanted to highlight one of my high school students that came in and gave a lot of time to us in actually performing some behavioral experiments where she was able to showcase that there was an age-dependent behavioral phenotype through a negative geotaxic assay. And so flies are normally negatively geotaxic. So if you hit this graduated cylinder, the flies will go to the bottom, and then they will immediately go to the top because they naturally go against gravity. They'll fly to the top. And so we can use this experiment to examine whether or not the flies are behaving normally. And so in this experiment, we compared wild type flies to our G11 severe fly and our D87A moderate fly, and we find immediately that in an age-dependent manner at day 4, our severe flies are unable to fly, climb, or walk, and our more moderate flies are able to actually fly to the top, with some of them not being able to. So again, we were able to find in an age-dependent manner that these PCH1B linked mutations actually exhibit these behavioral phenotypes in an age-dependent manner. And so, like our previous experiments, we wanted to understand, well, was there a role for this in normal neuronal function? And so we went back and looked at the mushroom body as shown here. And so in our RP40 CRISPR engineered wild type flies, we see no mushroom body defects essentially. However, when we look at the more severe mutants, we do see this beta lobe fusion or encroaching upon each other phenotype that we saw in our initial experiments, underscoring that the RP40 subunit of the RNA exosome is indeed required for proper neuronal proper mushroom body function. And these data start to build the case that RP40 may be required for proper axon guidance and neurite extension, given that we're seeing these beta lobe fusion defects in our flies. And so to wrap up this part of my talk, we are also, I wanted to look, kind of summarize the organismal defects. So first, we're just looking at gross organismal defects. Are the flies exhibiting any gross morphological defects, such as behavioral? Are there any defects in the brain, et cetera? And we were able to demonstrate the reviability, shortened lifespan, also behavioral defects, as well as mushroom body defects. However, we are still thinking about this molecular machine that is essential for fundamental RNA metabolism. So what is happening at the molecular level that is giving rise to this disease? And more, a better question, why are two mutants with single amino acid substitutions behaving differently? Why is there a spectrum in disease severity? And so again, our model is that in a normal individual, the RNA exosome is degrading and turning over RNAs normally, but in an individual with PCH1B, that this function is impaired. And so what we did is we did an RNA-seq experiment where we can actually look at all of the RNAs in a cell of the fly, and we did this by enriching for a neuronal population by taking fly heads and then looking at the fly head transcriptomics. And so the question we're asking here is, what are the altered activities towards specific RNAs when this subunit is mutated or defective? And so our first set of data where we were able to utilize our RNA-seq was to demonstrate that using PCA unbiased principal component analysis, we were able to understand that each fly, the wild type fly and the two mutant flies, had distinct molecular signatures. And so I'd like to impress upon the audience that, again, these mutations are missense mutations that are encoding single amino acid substitutions. So the difference between these different populations are single amino acid substitutions, and we were able to find in our PCA analysis of our replicates that indeed each of these flies had distinct molecular signatures, which gave us a clue or some insight that this may be why we are seeing the severe phenotypes that we're seeing, and also the differences, given that the mutants are clustering away from each other and also clustering away from wild type. And so next we wanted to look at what actually is happening. So the RNA exosome is a decay machine, right? And so if the RNA exosome is not functioning properly, what is happening or what should happen to RNA targets in the cell? If the machine that's really important for degradation of RNAs is not functioning properly, you would see increase in steady state levels of RNA transcripts in the cell. And indeed, that is exactly what we saw in our mutant flies, underscoring that our model was indeed modeling an RNA exosome defect, given its primary role in RNA decay. And so here in our volcano plots, we are showing increased steady state levels in both mutants compared to wild type in both cases. And we were actually able to see critically important RNAs for neuronal function in these sets of data. So ARC1 is a critical RNA, functionally important RNA for neuronal function, and so is this odorant receptor that we also saw. And what's actually fascinating, that in the more severe mutant, we saw the magnitude of the critically important RNAs for neuronal function increase as compared to those that were in the more moderate genotype, giving us a clue as to why, or molecular basis as to why we may be seeing these differences in genotype, or why we may have these genotype-phenotype correlations. And so, of course, we were able to validate these results via qPCR. And here we have an IGV viewer of ARC1 showing the actual levels of ARC1 in both the G11 mutant and the D87 mutant compared to wild type. So these are the real actual reads in our seq data of ARC1 in G11 and D87 compared to wild type. And of course, we validated that via qPCR. So this is really setting the stage for why is it that the RNA exosome may be critically important for neuronal development and function, given that it is regulating these critically important RNAs that are important for neuronal function. And so to wrap up and end my talk, I wanted to summarize kind of the molecular defects, where we talked about organismal defects and now molecular defects, as this complex is a multi-subunit molecular machine important for fundamental RNA metabolism. And so we saw that consistent with the primary role of the RNA exosome in RNA decay, that the majority of the gene expression changes detected in our mutants were increases in steady state levels. And also this revealed increased levels of key neuronal mRNAs in the RP40 mutant flies, underscoring that the RNA exosome may be indeed important, critically important for regulation of key neuronal mRNAs critical for neuronal function. We were able to do some rescue experiments with the human XOSC3 in these flies, and we were able to rescue these ARC1 level, these ARC1 increases in steady state level, but I'm unable to share with you those experiments because of time. But I did want to put that out there. And so now to our overall model. And so here we have a wild type RNA exosome that, as I mentioned very early on, that cofactors are delivering these RNA substrates such that the RNA exosome can process RNAs and degrade them. And then also, as I mentioned, they are thought to be very critical for restraining the RNA exosome as well. But what is happening in the mutant RNA exosome? So we have a defective subunit here where we have these missense mutations encoding these single amino acid substitutions. What's happening? What could be the model? These defects in the RNA exosome could lead to levels or complex integrity. So is the RNA exosome assembling properly and deassembling properly? Is it able to also let cofactors dock onto them with these defects? Are cofactors able to deliver substrates as they normally would? And then altogether, whether it is levels or complex integrity or these cofactor interactions, they all would merge to alter activity towards specific RNA targets, as demonstrated here. And this would be the crux of the model. So either way, whether it's cofactors or levels, complex integrity, this would alter activity towards targets of the RNA exosome, ultimately leading to disease pathology. And so very quickly, as I am a brand new PI at USC, I wanted to talk about some things that I am embarking on post my postdoc. And I am now interested, as I mentioned, in thinking about the RNA exosome, and now that several subunits of the RNA exosome have been linked to disease, I am interested in systematically investigating each of these mutations and in the subunits to understand what is happening and how is it that these subunits are giving rise to distinct disease. And so we will be comparing the different missense mutations that are encoding these single amino acid substitutions in the fly. And now I'm also adding something to my portfolio. We will be using stem cell derived cerebellar organoids to also model these RNA exosome mutations to understand what are the cell specific requirements of the exosome in cerebellar cell types. And so we will be looking at this to identify distinct and common phenotypes, again, both at the organismal or cellular level, and also thinking about what are the molecular profiles. And so in acknowledgement, my lab is small now, but we are growing. I have a postdoc that is starting in December. Sam Ali, and then also have a rotation student, Nina. And then I have Anuti Seth, which this is as a picture of Anuti and me. Anuti is the first member of my lab, and we are actually ordering equipment. CO2 machines actually were delivered minutes before I got onto this talk. And so we are ordering equipment and getting ready to do our initial experiments in the lab. And so, of course, a huge thank you to all my collaborators at USC, at Caltech, and also at IU. And so with that, I think we can open up for questions.
Awesome. Thank you so much. That was a really awesome talk and lots of information, and definitely coming from different perspectives, making it accessible to the general public, but also it's so cool to see some of your work in this way.
Thank you.
Yeah, anyone have any questions that you want to type in the chat, or feel free to also unmute and ask them.
Hi, I'd like to ask a question. My name is Brad. I'm a bioinformatics scientist at Genentech. It was a great talk, really interesting.
Thank you.
I was wondering if you could talk a little bit about if you're thinking about, families who have kids who actually have this disease, like if there's any therapeutic angles or what it's like for them. And I think this slide's really interesting because it looks like if you could just upregulate exosomal activity, you could provide some benefit to the patients. I'm wondering if you're thinking about doing screens or what you could offer for people who have these mutations.
Yeah, so great question. So now there are no therapeutics for this specific disease. So it's really palliative care that have for these patients. And unfortunately, a lot of these patients do have early mortality. And so actually what happens is that these infants are born and they go home normally. So the parents don't know that there's an issue, and then they start to miss developmental milestones and come back to the doctor, and they started to do a bunch of radiographical tests and found this brain development issue. And so there are currently no therapeutics for this disease, but for me, I think what's important is that understanding the fundamental biology of the processes and how this gene functions will eventually lead to therapeutics. And so this has already been done with some of the an RNA splicing event that's defective in another disease, SMA, where they actually utilize the fundamental biology to make their therapeutics since it was a splicing defect. And so our hope, and in my group, is that the fundamental biology that we learn about the RNA exosome and its tissue-specific function actually helps development of therapeutics in the future, or serve as the foundation for those therapeutics.
Thank you, Derrick.
Great questions. Anyone else?
I've got a question. Hi,
Sure. Yeah.
awesome talk. I'm Adam Reitzel from Charlotte. Really excellent talk. I'm a biologist.
Okay.
And so I was wondering, you were emphasizing the similarity between yeast and Drosophila and human. And I was wondering about if in your reading or in your other research, if you've found organisms that have done away with this or in any way modified it greatly, that they found a way around to deal with RNAs.
So no, excellent question. And actually, there are great questions, evolutionary questions about the RNA exosome. So the current RNA exosome, and I mentioned this catalytically inert core, the six-membered ring, which is catalytically inert, actually has pH-like domains. So at one time, those domains had catalytic function. And so why is it that it doesn't now? So this seems to be a relic of evolution, that at some part that these actual members did have catalytic activity. And there is in some organisms such as archaea, different compositions of the RNA exosome. So its 10 subunit core is actually conserved, but it can be made up of like heterodimers such as RP41 and 42, and the cap subunit is actually all one subunit. There's 3 subunits, but it's all XOSC2. And so there are in evolution different makeups of the RNA exosome, even though it conserves its barrel structure and core, but also there are great evolutionary questions about why certain subunits still retain these domains that would likely lead you to believe at some point that they had catalytic function that they don't have now.
All right, thank you.
You're welcome.
Callis in the chat would like to know if there's anywhere that someone could read up on your research, like a website.
Yes, so I actually have not developed my own lab website. That will hopefully happen sometime in the near future. But if you PubMed me, my most recent paper was on the RNA exosome. I've written review articles and book chapters on the RNA exosome. And so you will be able to find out a bit more about my research in those papers. And hopefully soon we have a lab website and talk a bit more about what we're doing in the lab.
Awesome. Any other questions?
Hi, I'm Melissa. I'm a grad student and I actually work in the cerebellum as well. And I'm curious about your work with the cerebellum organoids. Like, we haven't done that in the lab, but it's something that we've been thinking about. And in terms of just trying to model how like the cells work together and things like that. So are you using it more to get like the cell type specific interactions, or can you just talk about that a little bit more?
Yeah, I would love to. So this is new for me, and I'm so excited about this project. And actually, a new Seth that's in my lab, I actually recruited from Georgia Quadrato's lab here at USC in the Stem Cell Institute, and has expertise in cerebellar differentiation, and will be heading up that project in my lab. And correct. So what we're interested in, the RNA exosome, as I mentioned, is ubiquitous. And so why is it that these cerebellar cells or the cell types of the cerebellum are hypersensitive to the RNA exosome? And so I conveniently always leave out that the fly doesn't actually have a cerebellum, right? And so we are interested in understanding the fundamental biology of the RNA exosome, which the Drosophila model is a great model for. But now I'm interested in actually looking at different cell types that make up the cerebellum and understanding what is the requirement for the RNA exosome in these cell types, because it's very interesting that in all of your brain with the different tissues, this exosome is mutated, right? It's mutated in all your cells in your body. So why is it this very tissue-specific cerebellar phenotype that's occurring? And so by using this cerebellar organoid model, we can get at what is going on developmentally with the cerebellum. And so we plan to do very, nice timed experiments, developmental experiments, looking at edited stem cells, which have the patient mutations, which will differentiate into organoids, and actually following along developmentally to see what is happening to the cells with this defective RNA exosome.
Just to follow up on that, do you think that it's something like on the epigenomic level? And if so, like what assays do you plan to use with the time course model?
Sure. So what what I speculate or we hypothesize is that certain cells, so there could be unique function required of the RNA exosome. So in post-transcriptional regulation of RNA, cells may require unique function of the RNA exosome, or the RNA exosome may exert specialized function in particular cell types. So the post-transcriptional regulatory processes that occur in certain cells may require a spectrum of RNA exosome function. And perhaps in the cerebellar cells, that the RNA exosome has a high requirement for processing at certain times in development, to where perhaps RNAs that need to be turned over, which have been demonstrated in other groups in developmental studies, that certain classes or RNA molecules have to be turned over before progressing into the next stage of development. And perhaps in the cerebellar cell types, the RNA exosome could have unique function in those cell types that it no longer can exert, given that it's mutated. Or it could be that there are specific transcripts that need to be turned over that are accumulating, of course, allowing for viability because you get a patient, right? But then not able to maintain normal neuronal function in that tissue. And so those are kind of my two thoughts at this juncture. And it is quite clear that there is a hypersensitivity to these RNA exosome defects in these particular tissues. And so we are interested in following up on that. And I believe the cerebellar organoid model will help us get to some of those answers. And so you asked about what are some of the experiments we plan to do. We plan to do bulk RNA-seq and also single cell RNA-seq to ensure we actually have cerebellum, and two, what is the molecular profile of these cells? So that will give us insight into whether the RNA exosome compared to wild type cells is supposed to exert some specialized function in those cell types.
Thank you.
You're welcome.
Awesome. Well, so many great questions and conversation. I definitely have more, but we are out of time today. So I just want to thank you again for the talk, and thank you everyone for coming today. If you have any more questions for Dr. Morton, feel free to reach out to him, and we'll have this recording posted as soon as possible so that you could rewatch it or share with others. And yes, our next talk is actually January 20th of next year. So we hope that you've enjoyed this semester of our 2 talks. But thank you again, Dr. Morton, and we'll be in touch.
All right, and again, thank you for inviting me.
Thank you.